Connaissance,
recherche,
sciences,
et sciences mathématiques,
théorie,
physique, philosophie,
conférence, archéologie,
environnement, nature.
First of all, I want to thank Jorge Cubo and Michel Laurent for having me invited and giving our small, very young group of paleobiology a chance to present our work here. But I want to thank especially Armand de Ricqlès because some years ago, when I was very interested, and I'm still interested, in the evolution of life histories, I decided to make a bone histology on fossil insular forms because it is, I think, one of the most important tools to understanding and to extracting the life history of these animals. So I contacted him and I sent him, we exchanged some emails on this topic, and he encouraged us very much to go on with our work, and he made a very, very important remark. He said, insularity may have intensive, in fact, enormous ecophysiological consequences, very likely to be reflected in bone histology. And he wrote that histological comparisons with continental populations should serve as a comparative standard to test this. This is what we did, and indeed, he was quite right with his predictions. So this is our modest contribution to this homage. Well, first of all, I present the research problem. So it deals with evolution of life history traits. It is a very important topic that does not only affect biology per se, but also many fields that are very close to biology, but not exactly biology. So understanding of the evolution of life history traits would provide important insights in other fields such as ecology and conservation, biodiversity, physiology, aging, evolutionary medicine, etc. And I certainly forgot some other very important areas. So at the beginnings of the last century, many people were aware of this importance of life history research, although at that time it was not called like this. So some people started to proposing mathematical models, demographic models, that aim to explain the evolution of life history strategies. The problem is that those models which are developed in most recent years are actually opposed with predictions from certain classical theories. So here we saw the possibility to test both approaches, the modern one and the classical one, using bone histology and applying it to fossil bovids from continental and insular ecosystems. A little bit background, because it's important to understanding what I'm presenting here. Darwin, as we all know, had a very important problem because he could not explain which mechanism actually underlies the process of natural selection. And it was not until the beginnings of the last century that some geneticists, Fisher, Haldane, and Sewell-Wright, started to combine Mendelian genetics with the Darwinian theory of natural selection. And this was a very, very important step in understanding how natural selection works. And they created a new area, which is population genetics. And especially outstanding was the work for us, for me now in my work, of Sir Ronald Fisher, who elaborated a mathematical model of population growth that became commonly known as the Malthusian growth model. So he found out that exponential growth was based on a constant rate of production of offspring in an age-structured population, and key to his model was the intrinsic rate of population growth, and he gave it the name the Malthusian parameter R. So nowadays we have two theoretical approaches to population dynamics that predict demographic trends, and they include environmental cues to understanding what's actually happening. So on the one hand, we have first the classical R-case selection theory. This is a line of thought that started actually with Dobzhansky in 1950. He found that the Fischerian model did not really work in tropical forests. So he could not really find that population growth was exponential in these areas. Later on, Cody made a very important contribution working on clutch size, on continental and insular forms, and he found out the same, that there are important differences, and the Fischerian model does not explain these differences. It was but MacArthur and Wilson who really tried to formulate this new theory based on the old Fischerian model in order to include these new lines of thinking. And Pianka extended later on this. He was very important because, very influential, because he gave names to the traits which should be either R-selected or K-selected. So what did they find out? They found that resource availability is a very important factor in population growth, and it is limiting to exponential growth. So they incorporated the effects of population density, what they called environmental carrying capacity K, into the traditional Fischerian model. And as every good mathematical model, they could make predictions, and their predictions were that on islands there is a shift from a density-independent R to a density-dependent K regime. Or in other words, the intrinsic rate of growth of a population, R, declines under high densities. So the key prediction, in other words, would be that under resource limitation, fitness increases through a decrease in reproduction rate. Well, very opposed to this idea appeared in the 90s James Brown with his metabolic theory, which is actually not a new idea, not a new theory, but it's the Fischerian model where he incorporated body size scaling. So this theory is based on the fact that life history traits scale with body size. It is actually not a new mathematical model, but he also makes predictions, and they are completely opposed. So he says that under low extrinsic mortality, which is a trait of insular ecosystems, fitness increases through an increase in reproduction rate, not a decrease. So we have two diametrically opposed theoretical approaches to what might happen with life history strategies on islands. We have, on the one hand, the Fischerian and later the Brown model with a J-formed exponential curve of unlimited growth and investment in reproduction, while the MacArthur and Wilson model shows an S-form logistic curve of limited growth, where population reaches carrying capacity, and after a certain number of generations, there takes place a shift from investment in reproduction to investment in growth and maintenance. Well, our approach to test these two theories is based on life history. The life history is actually a sort of trade-offs between various life history traits. The basic idea is that energy is limited, so we cannot invest energy in everything like growth, maintenance, reproduction, and so on, but we must decide in a certain moment of our life where we will invest and how much energy we will invest. The key trade-off of life history theory is the trade-off between growth and reproduction. So this is just what bone tissue records, because it shows the shift from growth to reproduction, as well as other life history traits, in the moment when we find the outer cortical layer or the external fundamental system where growth stops and reproduction starts to function. So first of all, I would like to show how an insular ecosystem works. Insular ecosystems are extremely simple, so they are ideal for studying things. It's not just a chance that MacArthur and Wilson used insular ecosystems, and even that Darwin was very fascinated by them. They are a sort of small labs that run long-term experiments over sometimes millions of years, things we cannot do in labs. The first trait of an insular ecosystem is that it is resource limited. This is simply due to the small area of land, so resources are limited. You cannot escape and you have to live with it. The first effect of this is that those animals that need very much energy and are at the top of the energy pyramid or the trophic pyramid disappear. They cannot live there. So for the habitual prey, there is a release from predation. This is, in other words, a decrease in extrinsic mortality. On the other hand, there's also a decrease in the species number, because not all species are equally able to cope with these limited resource conditions, so the insular faunas are impoverished. This leads to a decrease in interspecific competition, and both the decrease in extrinsic mortality and the decrease in interspecific competition bring with it an increase in population density of the remaining species. This means also an increase in intraspecific competition, and here we have what we understand as density-dependent selection. So this is more or less a scheme how insular ecosystems function. And another very important point is that we used bovids. Bovids are characterized by a uniformity of their life histories. So they tightly scale with body mass. And so if we have a shift from growth to reproduction, which is recorded at the beginning, with the beginning of the external fundamental system, then we can use the body mass of a bovid to predict how his histological bone pattern should look like. Here, this is a diagram where you can see that the red points are, for example, age at first reproduction, the yellow points are gestation, the blue points are longevity, and the green points are calf weight, all very important life history traits. And they scale clearly, tightly with body mass. So a test with a continental sample shows, I found some data for growth for continental bovids. So it is habitual for bovids between maybe 20 kilos and some 50 kilos or a little bit more, that they have an initially very, very fast growth, then they stop growing and start a reproduction. And this is not only in bovids, wild bovids like chamois, or feral bovids like Ovis aries from Solway, or domestic bovids like Ovis aries, the Aragon breed, but also other artiodactyls like peccaries, or even other mammals like wolf. So we can conclude that adult bovids, more or less a little bit smaller than 50 kilos, would show first of all a fast-growing tissue without any line of arrested growth, because they start reproducing very early, and this will terminate directly in an external fundamental system, while adult, somewhat larger bovids may show one lug within the cortex, followed by again by another growth phase, and then the external fundamental system. Our continental bovids really show this. So we have, for example, adult Capra hircus. We see an internal lamella packet, and then where it started growing, the growth is quite fast. Here starts the outer cortical layer, and we see some lines of arrested growth there. So this animal may have weighted about 30 kilos or 20 kilos, and after a year or somewhat more, it started reproduction. We have here Gazella borbonica, and the image is nearly the same, only that here remodeling in the cortex is very advanced. How looks a little bit larger bovid is it's looking like this. This is adult Tragoportax gori from the Miocene of Piera, and it is about 80 kilos maybe, or even may reach some of them 100 kilos. And we see one lug in the cortex, within the cortex, again a phase of growth, and then the outer cortical layer. So this bovid followed the typical predictions for bovids with the start of reproduction. Well, now comes the surprise, prepared. This is Myotragus. An adult Myotragus. Actually, it does not look like a mammal, like a normal mammal. It is composed nearly, almost completely of lamellar bone that is interrupted by some slightly faster growing bone. And in this specimen, for example, we can count about 15 lugs or something like this. Most of the lugs are double or even triple. I don't know why. I do not know the reason why. It is very typical in Myotragus. And so we used a large number of specimens. We used about 150 specimens of Myotragus that we cut, and for comparison, about 50 specimens of continental bovids of 3 species. And here we have juvenile Gazella borbonica from the Pliocene of Laina in Spain. It is really very, very fast-growing bone tissue. The specimen itself is very young still. And if we compare it with a very small specimen of Myotragus, this is a femur of only about 4 or 5 centimeters, something like this. The adult Myotragus weighs about 20 kilo or more. So we can see here, initially, maybe until weaning, a very fast growth. Then we see a slow growth, an annulus with a lug, again slow growth, and a second line of arrested growth. So we conclude that this animal, although it is so small and so immature, had about 2 years of life. So we can make a sort of skeletochronology because we have this very rich sample from the very, very smallest animals which have a femur of only about 3.5 centimeters to large animals which have a femur of about 15 centimeters. So to our surprise, these very large animals, there are really an important number of them, do not even show the external fundamental system, and they do not show fused epiphyses. So they are actually still growing. If we put this together, we come, for example, to an age for this animal here of about 15 years, and it is still growing. We have also another phenotype, which is, they both overlap, but it seems to be that we have two phenotypes. So this is one which is growing somewhat faster, but it stops growth after some 10, 11 years, and it starts to lay down the EFS. So it starts obviously reproducing. We don't know why we do have these two phenotypes, and we either can contribute it to differences in the stratigraphy, in the age, stratigraphic age of these animals, though they come from the same site, but they come from caves, and stratigraphy in caves, it is very, very difficult to do. So maybe there are differences, time differences, which are very important with ecological changes. The alternative is that we have here male and female phenotypes. So now we can see how Myotragus is growing. These are femora. This is a newborn, actually very, very small. And this is about 1 year old, 2 years old, 3 years old. For 5 years, looks very, very much like this still. And then we have about 15 and more lines of arrested growth. And in some individuals, we even don't have the epiphysis closed. And these are adult animals. So on the basis of the skeletochronology, we can now see what's about the growth curve, and we can introduce Myotragus in this growth curve. And we see that it does not do the very, very fast initial growth typical of all placental mammals, and the plateau for the reproductive age, but it needs a long time to grow, and probably reaches sexual maturity not before 10 years or more. And this growth curve is actually much more similar to that of alligator than to extant and even fossil placental mammals of similar body mass. So our results suggest or our results are consistent with the prediction of the RK theory from MacArthur and Wilson, namely that under density-dependent resource limitation and low extrinsic mortality, natural selection favors investment in growth with an extended juvenile period and in maintenance with a long lifespan at the expense of reproduction. Thank you very much.